Okay, let's see osmosis now. By definition, osmosis refers to the movement of water through a semi-permeable membrane. To understand this, let's use some space here to compare between the diffusion and osmosis. We said diffusion, it's the movement of the particles. These are called the solutes. Remember the solutes from chapter 2? So diffusion refers to the movement of the solutes. And usually it happens from areas of high solute concentrations to the low solute concentration, okay? Across, of course, semi-permeable membrane. For osmosis, we are talking now about the solvent, which is, most of the case, it's water. And water, we can look at its movement in two ways. Either it's moving from areas of high water concentration to low water concentration, or from low solute concentration to high solute concentration. So we need to be very careful here when we describe the osmosis in terms of how the water moving. We can say either it's from the high water concentration to the low water concentration, or from the low solute concentration to high solute concentration. You have to mention what you are referring to when you are using high or low, when you are describing the osmosis. This is very critical, okay? Remember, still, osmosis is passive transport. Still, we are not using any energy. We are not using any ATP. To understand this, let's see these two images. Here we have an area of low osmolarity versus an area of high osmolarity. Osmolarity, you can think of it as the concentration of solute. So this is an area of low solute concentration, as you can see these red dots, and this is an area of high solute concentration. And here we have the membrane. The membrane is freely permeable. It means it will allow these solutes to pass between the two areas. So since this is freely permeable, then we will have diffusion. We understand that diffusion, the particles will diffuse from the area of high to the area of low. This is what happened here. These particles, some of these particles diffused from the area of high to the area of low across the freely permeable membrane. However, if we have the same situation, low osmolarity, high osmolarity, but the membrane now is selectively permeable. It does not allow these solutes to pass. So usually if it was diffusion, if it was allowed, these have to move into this area, but this is not happening. What happens is that the water is moving from the area of high water concentration to the area of low water concentration, or from low solute concentration to the high solute concentration. This is low solute concentration, this is the area of high solute concentration. So water moved from low to high solute concentrations. That is osmosis. And therefore we will see less water. We are missing some of the water here, and we see an increase in the water volume in this area. But concentration wise, it is equal now between these two chambers. If you think of this as juice, you can drink from this and you can drink from this, and you will not see any difference in taste. Here, if you drink from this side, it will be less sweety, for example, whereas after osmosis happen, both would have the same concentration or will give you the same taste. Why this is important? Because cells, as we saw earlier, are always living and surrounded by water, by fluidy solution. And within this fluid solution, there are particles, there are ions, there are salts. And inside the cell also, there is the cytoplasm, which is also fluidy. And also it has some ions, some solutes. So it's very critical for the cell that the concentration of these two fluids is equal, because we don't want the cell to lose or gain water beyond what it can hold. Okay? So therefore, based on the osmolarity of the two solutions, extracellular and intracellular, we have three types of solutions. We have the isotonic, we have the hypertonic, we have the hypotonic solutions. Isotonic, that is how the cells in our body living, or that is the type of solution the cells within our body living within when we have the same osmolarity, the same concentration of solutes between inside and outside. And with this case, there is no net movement of water. Water can go in and out, and this will not change anything for the cell. No net movement of water in this case for the cells living in isotonic solution. Think, for example, if you go to the hospital, they will give you normal saline. This is like a salty fluid. Why they are giving normal saline, not water? Because this normal saline has the same concentration of salt as our plasma. The second type is the hypertonic solution. Hyper, as you know, it means high or above, and it refers to the solutes outside the cells. So outside the cells, this is the cell, this is outside, we have high concentration of solutes, extremely high comparing to inside. These solutes will try to come inside, but they will be denied by the plasma membrane. The plasma membrane will not allow them. But inside we have the water. The water will start to leave. Why? Because water is going from the area of low solute to the area of high solute. So in hypertonic solution, water would leave the cell, and this will cause the cell to shrink, and of course it's going to dehydrate and die. This is what will happen in hypertonic. Remember, hypertonic, it refers to the increased osmolarity or high osmolarity outside the cell. The other type is the hypotonic. Hypo means below, and still we are talking about outside the cell. So outside the cell, we have low solute concentration comparing to inside. Inside now has more solutes. These solutes will try to escape, but they will be denied also by the plasma membrane. But outside we have water. Now water would go to the area of high concentration, of high solute concentration. This is the area of low solute concentration or low osmolarity. This is the area of high osmolarity. So the water would try to get inside the cell, and it will keep going inside. The cell would start to swell and eventually can burst, as you can see here. That's in a hypotonic solution. Now, why these terms are important, as we said earlier, because we need to make sure that our patients are receiving isotonic solutions. We don't want to cause them tonic shock, meaning that giving them like hypotonic solution or hypertonic solution, this will affect the blood cells. So I will stop here. When we come back, we will talk about the active membrane transport.